talk about the LHON Development Program and the LEROS clinical trial for patients with LHON. We're very happy that you could spend part of your day with us today. Just wanted to let you know a couple of things before we begin. I would like to remind you that we are recording this webinar right now for playback, and it will be posted later today on the UMDF LHON webpage and in our multimedia section on the website at umdf.com. You can get to the LHON webpage by simply typing in www.umdf.org/LHON. Following the presentations, we will have some time for questions, and I want to help you understand how you can be part of questions if you have some. If you didn't email them to us at news@umdf.org. We will be having a few speakers today, and there is a box on your page on this webinar that says Q&A. It would be up to the right-hand corner, and it's a question mark. And if you have a question, very simple, click that. It will bring up a box, type your question, and send it to UMDF staff, and we will make sure that it gets answered for you, time permitting. And with that, I am going to turn this over to our moderators, Dr. Phil Yeske and Lissa Poincenot. And Phil, you are first.
Hi, Cliff, and welcome everyone. Again, my name is Philip Yeske. I'm the Science and Alliance Officer at the United Mitochondrial Disease Foundation, responsible for managing all of our scientific initiatives, including our grant program, an expanding scientific portfolio of investments in infrastructure that are all related to a very important project that we've begun within MDF called the Roadmap. And we use our roadmap to align all of our goals, and they're really focused on the three areas: even improved diagnoses for mitochondrial disease, develop treatments and cures for mitochondrial disease, and lastly, but certainly not least, optimize patient care for those affected by mitochondrial disease. Particularly, the LHON community is a very important part of the overall mitochondrial disease community. And so today, we're really pleased to offer this focused opportunity to the LHON community to learn more about the activity of Santhera Pharmaceuticals. They continue to develop an intervention for LHON. Now, today, also collaborating with the LHON project at UMDF, and we're joined by the director of that project, Lissa Poincenot. And Lissa, perhaps you could say a few words as well.
Sure, a lot. And thanks to UMDF and Santhera for being on this webinar today. For anyone listening to the call, I just want you to know that we have a very large, active, and welcoming LHON community. We've got over 3,500 people participating in our global LHON Facebook group. We have a monthly phone call the first Wednesday of every month, and anyone invited to participate. Next June will be the 6th time that we've conducted an LHON program in conjunction with the UMDF Symposium, where we focus on the unique issues facing the LHON community, which is sub-nonset legal blindness. And the UMDF is a great resource for learning more about LHON. As Cliff mentioned earlier, there's an LHON page that you can find on the umdf.org site. And there we store recordings of those monthly calls I mentioned and videos from the conferences that went so far. And there's also a link to LHON.org, and it has a link to our Facebook group. And you can also use its contact form to find me, Lissa Poincenot, and I'd be happy to help anyone who has additional questions and guide them to information resources about LHON. Thanks, Phil.
Sure, and thanks, too, for your collaboration. We really value the opportunity to partner with you on LHON-related matters and mitochondrial disease. We look forward to doing more programs like this, and certainly our annual symposium is always a great opportunity to bring the LHON community together as part of the mitochondrial disease community as we discuss basic research, translational research, clinical activities, and really support issues as well for the entire spectrum of mitochondrial disease. Well, what I do, I think we should move on to the meat of this, the content of today's webinar. Our first speaker is Dr. Xavier Loria. Dr. Loria has a medical degree from the University of Barcelona, where he has a wealth of experience, almost 30 years of experience in the pharmaceutical sector, from clinical development through registration of drugs and commercialization of numerous drugs. And the second half of this is really focused on the rare disease, orphan drugs and rare diseases. So he's currently serving as medical affairs manager for Santhera Pharmaceuticals out of Switzerland and responsible for Europe and the rest of the world. Today, Dr. Loria is going to talk about the Santhera Development Program for LHON and Rationale. So I'll turn it over to you, Dr. Loria.
Thank you very much. Good afternoon from my side. Good morning to you, and greetings from Europe. First, I would like to thank the organization for giving us the opportunity to share this webinar, and to technology for allowing me to be able to talk to you from this distance. Now, and also thank you very much for your kind introduction. Now, my job here is just set the scene and walk you through some data about the company and about the clinical development program of idebenone in LHON, in order to prepare the grounds for Diana, my colleague Diana Petraki, to inform you and update you on the LEROS trial. So now, the thing that I wanted to talk to you about is the company. We're talking about Santhera Pharmaceuticals, a small company. We are currently about 100 employees. We've just become, let's say, national. It's a company that was created back in 2004 by a group of actually scientific and academic professionals that were experts in neuromuscular and mitochondrial disease. Now, based on that, and with the belief that this could have an influence in the life of patients affected by mitochondrial diseases, they decided to found the company, which is what we now know as Santhera. And over the last 10 years, we've been busy developing idebenone for a number of indications that I'll show you now. And what I highlight from this slide is that since the approval of the indication for of idebenone for LHON in Europe, which happened a couple of years ago, well, obviously we had to expand operations and had to expand research, and luckily we've expanded it on the United States. So here that will allow us to boost both research and development in further diseases, also and mainly in LHON. Now, in terms of pipeline, in terms of what we do, right foundation, as I said, we were dedicated to focus on mitochondrial and neuromuscular diseases. LHON is actually a kind of well-defined mitochondrial disease that had no treatment, and that the company decided to further research the potential effect of idebenone in this disease. For the last 10 years, as I've said, we've been busy doing that, and finally it has culminated in the European approval under exceptional circumstances a couple of years ago. It is easy, as I said, we're a small company. It requires a lot of resources, a lot of risk, but we are happy to say that we've got there. Now we know more about the disease, and now we know more about the potential way of treating it. Now, that is not all. Obviously, you need more legs on the table. So we're also investigating in Duchenne muscular dystrophy. We are very advanced in that research. We've submitted the request for authorization in Europe as well. Investigating the role or the potential role of the idebenone in other interesting and rare diseases like the primary progressive multiple sclerosis. At the bottom of the screen, or in the middle of the screen, you can see the omega pill. That's another molecule which is being investigated for a rare, devastating disease in children, which is congenital muscular dystrophy. So, but that is in very early stages. let's change gears a bit. Let's move into the LHON. I know that most of you are familiar with the disease, and I hope you don't mind if I try to simplify this for the sake of understanding the disease, and later on position or tell you the role of idebenone in this disease. What I'm trying to depict here is that this is a disease that affects mitochondria. We know that. For a reason reasons that we still don't clearly know, it seems to have a preference for retinal ganglion cells. There are certain theories. The bottom line is that it affects the retinal ganglion cells, which are in the back of the eye. There is a structure that forms the optic nerve, which is going to transmit the image to the brain, transmit what the eye sees to the brain. Now, this mitochondria, we could make the symbol or similarity to a power generating station, and what we're showing here is that when everything works well, when the power station is working well, power is getting to our household, our appliances work, our TV functions, we see things clearly. So that's a normal situation. Now, what happens when the mitochondria, in this case, we make the image of this power generating station, doesn't work properly or doesn't work at all? That means that those cells that should transmit that signal finally will create the image in our brain. They are not working properly, which means that in the case of the power generating station, electricity is not getting to our households. We cannot use the appliances. Food is spoiled. We don't see TV. We cannot use the water pump, etc. So they're not functioning properly. And this is what we see in LHON. Due to that problem in the mitochondria, these cells are affected. As a result, we have a visual function deficit. So this could be the what causes and how the disease is caused. Another interesting observation, which is important to bear in mind, both for families and affected patients as well as physicians, is that this is a mitochondrial disease, and I've said that I haven't explained this, but the mitochondria has a genetic problem, and this is the problem that will ultimately will create this function. Now, saying the genetic problem in the mitochondria, it's important to highlight that mitochondria are passed to the descendants by the female, not by the male. So this is a disease that is going to be transmitted by the maternal line of the family, the characteristic of mitochondrial diseases. Now, another important thing to bear in mind is that not all people having the mutation will develop the disease. It seems to have to be more frequent in males more than females, but for whatever reasons that we still don't know, well, it seems to be more frequent in males. It's important that to insist that when you have a family in which you have a member affected, the family obviously has to seek advice in terms of lifestyle and what drugs to avoid, etc., in order to prevent as much as possible these mitochondrial problems being worsened. As I've said, another important thing is that males don't pass the problem to the descendants. Now, let's change page here. Let's talk a little bit about idebenone. What is the rationale? Why we think and why we started developing it in the LHON. On the left side of the screen, you can see what we've already known, that this is a mitochondrial disease. It affects the retinal ganglion cells. And that an important is that these retinal ganglion cells don't work properly, and they lose function. And this function might be lost for a certain period of time. And we talk about viability, saying that cells, even if they are affected, they can still be viable, and they can still at some point have the potential to recover the function. How long? That's a difficult thing to say. We are researching, we are developing, we are getting new knowledge, which all this, what we considered that traditionally people would say that, over 5 years or past the 5 years mark, a number of recoveries is not very important. Well, now we might be changing that. So that's something that is changing with time as we learn more. And another important thing that I wanted to highlight is what we call the window of opportunity, which means that due to the nature of the disease, that you have cells that lose function, but they are still viable. That gives us a chance to try to recover the function of those viable cells, those viable structures. During this period of time that I'll give you details in a minute, we could try by therapeutic approach to recover that function. Now, the way that Santhera approached this problem was by looking at different drugs, different products, knowing that the base of this disease is a problem in it's very, very well-known problem in the mitochondria, and specifically in the complex 1, which I'm not going into detail with that. The company knew that idebenone could have the perfect profile to tackle this problem in terms of its antioxidant and the capability of shuttling electrons across or along this respiratory chain in the mitochondria. This is too complex, but just for you to know that these two effects would be the basis of this use of idebenone in this disease. I want to highlight that, as I said, the drug is approved in the European Union under exceptional circumstances. That's due to the fact that this is a rare disease, so we cannot get a lot of information. So with the information we've got, we know that there is efficacy and safety, and the authorities allowed the drug to be available for use in the public. Now, the clinical dose that has been used and the one that is approved is 900 milligrams a day in 3 doses. This is important, and also very important to take it with food because it's a drug that has difficult absorption. Before I forget, I want to mention that this drug is not approved in the United States. It's still investigational. So that is due to the different criteria or requirements in the European setting and in the North American setting. Now, having said that, and I know that I have the risk of oversimplifying this, idebenone could be made similar to an emergency tire in a car. The function of a car is to move us from point A to point B and back, and carrying on moving us along. Sometimes we have a problem and we puncture a tire, or a tire runs flat. So then what can we do? We can get stuck and not move. That would be a situation in the mitochondria, LHON, or we could try to, even if it is with something that is not 100% this is the original, we can try to substitute. This is the emergency tire you can see here. That allow us, obviously with some restrictions, but it still allow us to be mobile and to perform the function of taking us from point A to point B. And this is a bit what idebenone would do in the mitochondria. It would help the mitochondria function, even if it is not through the substituting the. So this is important. This is not a cure. This is just a drug that tackles one of the problems. I've mentioned about the window of opportunity, and this is what we're trying to see here. This is a disease that evolves very rapidly, as probably, unfortunately, many of you know. But even if when you reach that loss of visual function, you still have cells that are viable. And when those cells are viable, there is the opportunity of treating. This is why we say here that we try during that period as much as possible to recover. And in the case that we're talking, we use idebenone for that. In terms of this disease, we know, as I said, that it evolves very rapidly. Normally, it stays with this loss of visual function. In some cases, as you probably know, there might be some what we call a spontaneous recovery, and occasions through therapy we could try to provide benefit. And this benefit, and you will see later, we measure it in terms of both things. One is recovery, so to regain lost vision, or up to a certain degree, of course. And the other one is stabilization, and we mustn't forget about stabilization. Stabilization is trying to prevent further loss of function, which has to be seen also as an important therapeutic objective. Now, just to move to the second part, I want to show you the program. This is the clinical development program. I know it looks very busy, but let's simplify it in the following way. First, because this is a drug for a rare disease, we were given orphan drug designation back in 2005, 2006, and with that we started the clinical development. This step was the clinical trial LEROS, which we are happy to say that was actually a very important achievement. It's a clinical trial, randomized, double-blind, placebo-controlled, and this is the first one that was done and published in LHON. That gave us information that allowed us to obviously see what the potential efficacy and safety would look like, and that allowed us to move us to the next step, which was gathering further information in real world with the dose that I've mentioned to you, 900 milligrams a day, in a real world setting, as I said. But that is not enough. Another step that we needed in order to get approval was to better profile the disease, and that's the reason why we performed this last study before approval, which was what we call clinical record survey or natural history. So in order to know better how the disease evolves in the natural setting when it is not treated. This is what we depict here from the clinical trial. We get information. Unfortunately, the clinical trial only lasted 6 months. So if we want to know what happens or beyond 6 months, then we went into this expanded access program, which provides us more information, and the natural history us to put all of that into context. I want to highlight one important thing for such a rare disease like LOM. We've got data from over 500 patients, not all of them treated, but 500 patients providing data on natural history and efficacy data. Now, you will hear during my colleague's talk, Diana's talk, about these criteria, the clinically relevant recovery, the clinically relevant sterilization. And what I want to say to you here is efficacy, what we've learned from all these studies that we've done is that probably the best way to measure efficacy is by looking at these two things that I've mentioned. Recovery on the one hand and deterioration on the other. Now, this is measured through visual acuity, and probably most of you are familiar with these charts that we are asked to tell the letters, etc. So that's how we measure visual acuity. The idea in recovery is to see improvement, but we want to look at that from what we call a meaningful, relevant improvement, something that really makes a difference. That's what we say here. So improving 2 lines or 10 letters on this chart is considered to be relevant, or going from off chart to on chart is also considered to be relevant. So that is what we know as clinically relevant recovery. And when we want to have a look at the prevention, to avoiding deterioration, that's what we look at through this clinical relevant stabilization, which in very simple terms, I'm not going to elaborate more on that, is basically that you try to maintain visual acuity within a certain level and not deteriorate further. Now, with all of that and with all the data that we've gathered, we still have questions that we haven't been able to answer, and this is the reason why we are doing further studies. And this is the reason why LEROS is here, and this is the reason why we have other studies even after authorization. It's what we call post-authorization measures, which I'm showing you in this slide. The idea is that we need to get, or we want to get more information that can allow us to answer questions like what type of patient that responds better, how long does it take to respond, how should we treat, what is the safety profile in the real world, do we have data about the natural history. So this post-authorization measures is something that we are engaged doing and informing our European authorities on the ongoing results. Part of those measures is a LEROS study, which is what Diana Petraki, my colleague, is going to tell you about. And from my side, that's it. We will have time to answer questions at the end of this session. So far, I want to thank you for your attention. I want to hand over to my colleague, Diana, I manage to do that.
Thank you, Dr. Loya. Maybe while
Thank you very much.
That over, I'll quickly introduce Diana Petracchi, and thanks for that very informative presentation. Diana Petracchi has a master's in drug development. Has been working for Santhera now as a clinical trial manager for what sounds like almost 9 years. Also very experienced in development, working at clinical research organizations before joining Santhera. Is going to talk very specifically about the LEROS study and how it is being deployed for further testing of idebenone. Diana.
Thank you for the introduction, Phil. So as mentioned, I'll be presenting the LEROS trial. So let's get started with the study design. So it is an open label study, which means that everyone, essentially what we mean by open label is that everyone knows what treatment they're receiving. So LEROS trial, all patients that are eligible to participate in the trial would be receiving idebenone, so there is no placebo. The study is being conducted in the U.S. in 9 sites, which we'll go over a little bit later, and also in Europe in 22 sites. Looking to enroll a total of 250 patients with 125, so half of those patients having their onset of symptoms less than a year ago, and the other half, so 125 patients having their onset of symptoms between 1 year and 5 years ago. So study objectives, essentially what that means is what we're trying to achieve with the study. So first, we want to evaluate how much idebenone helps recover or stabilize vision for patients with LHON for less than 1 year and more than 1 year since their onset of symptoms when we're comparing to an external natural history control group. And we're also looking to evaluate the long-term safety of idebenone with LHON patients. So measure this, that's what we mean by endpoints. It is, as explained by Dr. Loya before, so we look at how many eyes have a clinical relevant recovery. We compare visual acuity, so the of the vision at visit 1 compared to 1 year after treatment. And we'll also compare that to the natural history control group. Now, what would be required of participants if they were eligible to participate in the trial? So they would take idebenone tablets 3 times per day with meals, so that's a total dose of 900 milligrams per day. And that intake of idebenone would need to be recorded in a patient diary. And that patient diary would be distributed to the participants at each study visit, so they would receive that. And we'd also record things such as any side effects or any medications taken between visits. So participants would come to the clinic a total of 9 times over 25 months. So lower on the slide we have on a timeline the interval of those visits. B refers to the baseline visit, which is visit 1. And after that visit 1, participants would come back at 1 month, 6 months, 12, 18 months, 24 months after that baseline visit or that visit 1. And the final visit would occur about a month after that. So that's a final safety visit. But the treatment period would be 24 months. Visit 1, once it's been confirmed that the participant is eligible, they receive drug to take home. And, typically, what we've been seeing so far is the labs that are, so the analysis of the laboratory results isn't necessarily in on the same day as visit 1, and some of those results are necessary to confirm whether the participant is eligible or not to partake in the study. So normally what we see is the patients get a call from the site to say, okay, it's okay for you to start taking drug. We've got the labs back. Everything looks fine. So that's typically how it happens. And after that visit 1, there would be regular assessments in the clinic, which I'll go over in more detail in the next slide. I'll visit 8. And then, as mentioned before, there'd be that safety follow-up visit 20 to 35 days after study medication is stopped. So the criteria, what that means are the criteria or the specific conditions that need to be met for a patient to be eligible to participate in a trial. So for the LEROS trial, it's patients that are above 12 years old, so 12 years and above, have had their onset of symptoms less than 5 years ago, have one of the 3 main mutations, which are the 11778, the 460, and 14484. Women patients can't be pregnant, and they must be willing to use highly effective contraceptive measure until the end of treatment. The exclusion criteria, so these are basic conditions that should not be met for people to be eligible. And in the LEROS trial, those criteria are any previous use of idebenone in any form would disqualify a patient from participating. If there is any cause of visual impairment, so examples of that are macular degeneration, cataracts, glaucoma, so anything that in itself could also be causing a vision loss. Under that same idea, there's also any active ocular disorder that would exclude the patient from participating, and then participation in a clinical trial. So that includes gene therapy trials within 3 months prior to participating in the LEROS trial. So here we go. What can be expected at the first study visit. So patients would receive an informed consent, and they'd be given enough time to review that and decide whether or not they want to participate. They would also be given time to ask questions. So all of that would be done with the site staff. And if a patient does decide that they want to participate, then they would go ahead and sign that form. If it hadn't already been done, there would be a sample drawn to do a genetic test mutation to confirm the patient has LHON, as well as additional tests, some eye tests. The doctor would go over the medical history with the patient, make sure that they meet the criteria that we reviewed before. So there are more than the criteria that I listed on the earlier slides, but those are main ones that can help patients identify whether or not they think they'd be eligible in the trial. They'd also review all medications that patients are currently taking to see if there's in there that would exclude participation, but that's just things that are done in clinical trials. There's also some safety bloods that are drawn, so as I mentioned, to confirm whether or not the patient's eligible, and also a urine sample, be a pregnancy test for women, and a visual acuity test, so to measure the visual function of the patients a physical exam, so the doctor would do a physical exam and also an exam of the eye fundus, which is referred to here as a fundoscopic exam. And the end of the visit, so once all of this is done, the patient would receive study medication and a diary, which I mentioned before. So at the study visit, a little similar, but a little bit shorter than that first visit where a lot of tests get done. So this visit, the study team would review with the patient compliance with this medication. So if that errs on the side of being too low, then that would be rediscussed to remind the patient to take their doses and that sort of thing. They would go through the diary to see if there have been any side effects experienced between the visits, and if there's any new medications that were taken since the last visit. It would be the safety blood and urine test, so that's standardly done at each visit, visual acuity test as well, and then physical exam and measurement of the vital signs, and that one is not done at each visit. That would be the distribution of the study medication and diary again. So here's the distribution of the sites across the U.S. So they're across the map. So this is just to give you an idea of where they are. On the next slide, I'll go over are located exactly. So more information that can be found on clinicaltrials.gov if ever you want to refer to this information later on. All the sites are located also for Europe. Site in New York, a site in Bethesda, Maryland, a site in Phoenix, Arizona, Mountain View, California, Palo Alto, California. So there's two sites closely located in California. Atlanta, Georgia, Charlottesville, Virginia, Aurora, Colorado, and so, thank you for your attention, and also if there's any questions that come up after this webinar, you can always reach out to our patient advocacy group representative, Jody Wolf.
Diana, this is Phil. Cliff, would you like to kind of kick off the Q&A session? Or not?
At 20 minutes left in this webinar. If you want to ask a question, it's very simple. Just go to the Q&A box and select UMDF staff and then send us the question. We have some in, and I've been forwarding those along. And I don't know if you have any in that you'd like to ask on behalf of the patients.
Thanks for sending those along. If I can ask a couple of demographic questions that have come in. One question had to do with the minimum age of 12 years. Could you comment on why there is a minimum age?
You talk about the LEROS study? Yeah.
One, but I think the question was about the LEROS study, but feel free to comment either way.
Mm-hmm. Well, maybe you should take that question.
I don't have exactly the answer to that. I mean, I think typically trials so below the age of 12 would qualify as a pediatric trial, and those are separately. But I don't know. Yeah, if you have further.
Yeah, and if I further elaborate on that, I agree with what they says. It's basically because of the age considered to be a child and and not adolescent still. In terms of pharmacokinetics, in terms of other iterations, that there will be a limit in the sense of children behaving differently than adults, etc. So it's more related to the cutoff age of childhood.
Then another demographic question. Someone was asking, have you observed or can you comment on whether there's any difference in how men or women respond to idebenone?
Not really. No, the gender doesn't seem to be a factor that influences response. So we can say it's in the clinical trial as well as in the other programs that I've mentioned to you, men respond as females in terms of numbers and in terms of magnitude. So no gender influence.
Phil, did you want to toss in a question?
Sure. So there have been a few questions around the regulatory process in the United States. Perhaps Dr. Loria could speak to that process for approval of Rationale in the United States, either milestones of what you need to achieve or a rough timeline for Santhera's plans for further development of Rationale for approval in the United States.
Yeah. My knowledge is, as my title says, is more about Europe and the rest of the world, so I'm not 100% fluent on the FDA. What I can tell you is that we are engaged in conditions with the FDA in terms of requirements, in terms of the process, in terms of time. So that is ongoing. In terms of milestones, I don't feel qualified to give you specific milestones because I'm not aware of them, just to know that we are engaged in negotiations with the FDA. In the meantime, we are busy with the research, doing the LEROS study. It's still, in the regulatory process. Unfortunately, the health authorities in different regions of the world have different requirements. That explains also the difference in timings. Mm-hmm.
Another question, one of the participants was wondering whether idebenone should be taken in conjunction with other supplements. Does that impact efficacy in any way?
In order to give an answer to that, we should studies, and the studies have been done. No formal studies have been done, and we haven't done formal studies in the use of idebenone together with other substances. So we know, and that's what we can talk about, is about idebenone itself. So no, I don't have any, and I can talk about and you take it with other supplements. That would require clinical trials and obviously formal studies.
Fair enough. That's what I have from my side.
We've got a few questions about the actual LEROS trial. So maybe, Diana, these are for you, and it's going to be 3 different questions sort of before, during, and after trial. One question is thinking of participating in the clinical trial, and they don't live right next door to one of the centers that you identified. Should they be concerned about the cost of getting there, especially if they have difficulty going on their own?
So, yeah, we would cover for the cost of traveling as well as companion travel. Haven't considered, though, for reimbursing for patients. So basically just travel, if there's plane or mileage or any hotel that needs to be booked.
Great, thanks. Another question is, we have an individual who's a parent of someone who's currently involved in the trial, and I know you talked a little bit about, how the product should be taken, but could you elaborate a little bit more if they want to get like maximum absorption? Are there any tips, tricks, or recommendations on how they could, best, foods to eat or, specifics to take, or is it just more general than that? What would you say on that question?
I know that Dr. Loria is very well placed to answer that question because he's answered it before. Do you mind if I transfer that one to you?
Sure.
Yeah, this is Loria. In terms of administration of the product, I insist, and I always insist, it is important to take it 3 times a day, and that because the way the body handles the drug requires this sequence of 1, 2, 3 a day. That ideally has to be combined with food because the absorption of idebenone is not great in the body. What we've seen is that when you take meals, let's say a breakfast, a lunch, a dinner, together with the idebenone, that increases the absorption even up to 5 to 7 times the amount of drug that gets into the body. So that's why we insist, and I always insist on taking it with food. Now, you ask me what food, and I might get all the cardiologists against me if I say that, fat seems to be better than let's say, normal protein or whatever. But I'm not going to say that you have to take butter with it. But if you take a normal meal, what you would eat normally for lunch or for dinner or for breakfast, that would be enough. So don't need to take a special foods for improving absorption.
Thank you. And then the third question related to trial participant is, if an individual does take part in the trial, and let's say the good news is considerable improvement actually does occur during the trial, what happens at the end? Is there any chance they can carry on taking the product, or must they stop and risk losing whatever gains they've had?
It's on a case-by-case basis, as you mentioned. If there is an improvement observed, that would options to provide continued access.
Cliff, anything coming to you?
I had a few more. I just didn't want to hog all the questions.
No problem. Please continue.
Okay, let's see. Interesting question. Somebody's asking about UK's exiting the EU. Is that anything that's going to impact trials or use of idebenone? Anything to consider?
Wow, that is a really difficult question. I don't know. It shouldn't. But this is my very personal opinion. I believe that negotiations will happen. The UK Brexit, everything regarding health and regarding treatment and regarding approvals, etc., will probably be as it is now, in line, or at least overlapping one the other. So I don't think that the requirements or the things are going to change because of the Brexit event. But having said that, I don't have any evidence, or I haven't read anything about the plans that they might have.
Okay, and then somebody else is asking about LHON plus, and just wondering if there's any thoughts to doing any kind of a trial around the concept and the issues around LHON plus.
We don't have we don't currently any plans of doing LHON plus. Still busy, with this profiling the LHON. Clinical trials require a certain number of patients, population, and it is a huge kind of effort in terms of design, in terms of include patients, etc. So it's not an easy task, and it's something that we would need to think very careful.
And somebody else is bringing up the point that within patients who are sort of considered in that chronic phase, the question is about how some folks report that they see blackness and others report seeing a cloud of colors. Are you able to speak to whether this has any significance in terms of whether the RGCs are viable and treatable, or would you really be able to speak to that?
I would lie to you if I say that I'm qualified. No, I don't have an explanation for that. I have my own theories, but I'm not quite to really give you a proper physiopathological explanation why. Vision is something that happens, but at the same time is subjective. So the same as I believe that they report that they see the scotoma as black, other that see scotoma as white. To my knowledge, that can have a relevance in terms of viability. But as I say, I'm not an expert on this, so this is a specific issue or topic. Probably, I cannot comment on that.
Sorry,
anything more from your side? Mm-hmm.
I'm trying to read something while I'm also being on the phone. Somebody's asking about rare mutations, and I don't know that I can read the full question. I'll leave it with you. Have any insight on, or have you done any looking into rare mutations?
Yeah. What I'll tell you is what was presented recently at two scientific meetings in Europe. One was in Budapest, and the other one was in Nice, in France. About a month ago, more or less, and we presented some data on the few cases of rare mutations that have been in routine clinical use in different countries, and basically it was to inform on the findings. Nothing special. They seem to respond okay, but it's very limited the number of cases. And from that, obviously, you cannot hear or deduct any specific insight. What I would say is the message I would get from data is treated responded or seem to respond very well. But the message for me would be, if the suspicion of LHON, if clinically everything could or it could be LHON, and the primary mutations are not found in the genetic test, I think personally that it is really, really worth to look for other mutations, because even if it is a rare mutation, it benefits of potential therapy. So it's good to identify the mutation for whatever might happen next in terms of therapy, whether it is idebenone or whether it is another one that could be in development. So that would be my message. Don't stop at the 3 primary mutations. Go identify the co-mutation if that is possible.
Great. And in the U.S. there are people or organizations like GeneDx that do look for up to 20 mutations. So there certainly are ways to do that. That's good insight. Some are asking if you can just comment a little bit on what it is that Santhera is bringing with this product that differs from what people might be aware of on the internet today. Is there anything just generally you can speak about on that topic?
Let me just answer you with a question. Are you asking me about idebenone or about other therapies?
No, idebenone. Mm-hmm.
Idebenone. Okay. Now, this is a kind of tricky question, and I will try to answer it kind of slowly. What we bring in is a drug which has been subject to clinical trials. So for that, we had to develop a formulation. We had to develop a way of put idebenone in a tablet, and then that tablet with all the excipients and all the principal substance, which idebenone, etc. That is what was tested in clinical trials in terms of efficacy, safety, etc. Now that requires, in terms of health authorities, very strict controls in terms of making sure that the active substance is always the same. It's always present in the right amount. There are no toxic derivatives from the manufacturing process that sometimes in certain drugs happen, to make sure that the amounts and the full product is consistent and kind of guarantees that what you are taking is what has been tested and approved by the authorities. That is what comes with when you consider something a drug. Now, if you get idebenone and you say, well, there is idebenone on the internet, there is idebenone as a supplement, it's precisely that. It's a supplement. Supplements are considered to be safe by definition, but they don't have any, they haven't been subjected to tests in terms of efficacy and reproducibility of that efficacy, etc. What I'm trying to say with this is we know that when we produce our idebenone, we know that the amount of idebenone is that, and that is going to be tablet 1 to tablet 10,000 if you want. When you get products from other sources, that to the health authorities to check whether that is the case, whether that the active substance is the active substance in the right amounts, in the right with the right excipients, and no traces of potential toxic or other substances. They cannot comment, obviously. So what we bring is the confidence that the drug fulfills criteria of stability in terms of reproducibility of what the drug is, and that drug, exactly as you are taking it, is what was tested in clinical trials. That is what I can say. Further, what the supplements or what you find on the internet, that I don't have enough information. I cannot comment on that, and it wouldn't be fair for me to comment on that, honestly.
That's great. That's great. I appreciate that. Actually, a few questions have come in also about the clinical trial, specifically about LEROS. Diana, can you speak about, you mentioned there's 9 trial sites in the U.S. Are all of them active now or not yet?
Yes, they're all active. They're all open and active. We're updating clinicaltrials.gov just for that last site that got activated a couple of weeks ago, so they'll all appear as recruiting.
And then there's a question about does the individual who's interested in participating, must they go to the closest trial location?
I'm trying to understand. Would it go to the closest one? Is it because they're already followed at a certain center, I guess, and going there?
I'm just reading a question that came in, but I'll just imagine somebody might say it's easier to get somewhere. Huh?
I heard they go to the closest site.
Okay. And when someone does get there, I know you went through some of the tests that will be performed. Visual field testing is a real favorite in our group. Is that one of the tests you'll be doing on participants in the trial?
So there's the visual acuity. I didn't specify it actually when we were going over it, but it just a read of a chart basically on a fluid panel, and then yearly and once yearly they'd look at the back of the eye. So it's no field.
Great, thanks. That's all I'm seeing. Phil or Cliff, if you have anything else, please jump in.
I think we are almost out of time. We have about a minute left, and I just want to thank everybody for participating today, and just remind you that if you want to go back and hear the information again, you can go to www.umdf.org/LHON. We'll post this webinar there and also on the website. And again, thank you all for participating. I hope this has been informative for everyone.
Thank you much.
Yep. Well, thanks much for your attention, and have a good day and enjoy the rest of your day. Yeah.
Thanks. Bye.
Thank you.
Thank you. Bye.
Bye-bye.